Hello everybody, Kemo Sabi here, coming at you with another video. This one's off greenmediainfo webpage. I read it by Sair Jai Founder, published on October 28th of the year 2018. A little bit older, but still thought this one could be relevant. With the topic of why everything you learned about viruses is wrong. Groundbreaking research indicates that most of what we believe about the purportedly deadly properties of viruses like influenza is based on nothing more than institutionalized superstition and myth. Germ theory is an immensely powerful force on this planet, affecting everyday interactions from a handshake all the way up the ladder to national vaccine agendas and global eradication campaigns. But what if fundamental research on what exactly these pathogens are, how they affect us, has not yet been performed? What if much of what is assumed and believed about the danger of microbes, particularly viruses, has completely been undermined in light of radical new discoveries in microbiology? Some of our readers already know that in my previous writings I discuss why the germs as our enemies concept has been disseminated by the relatively recent discovery of the microbiome. For in-depth background on this topic, read my previous article, How the Microbiome Destroyed the Ego Vaccine Policy, the Patriety. You can also read Profound Implications of the Virome for Human Health and Autoimmunity to get a better understanding of how viruses are actually beneficial to health. In this article, I will take a less physiological approach and focus on influenza as a more concrete example of the Copernican level paradigm shift in biomedicine and life sciences we are all presently fully immersed within, even if the medical establishment is yet to acknowledge it. Deadly flu viruses vaccinate or die. The hyperbolic manner in which health policy makers in the mainstream media talk about it today, flu viruses, is a deadly force against which all citizens of all ages 6 months or older need to take an annual influenza vaccine to protect themselves against lest they face deadly consequences. Worse, those who hold religious or physiological objections or who otherwise consequently object to vaccinating are being characterized as doing harm to others by denying them herd immunity, a concept that has been completely debunked by a careful study of evidence or lack thereof. For instance, in an interview, Bill Gates tells Sanjay Gupta that he thinks non-vaccinators kill children. But what if I told you that there isn't even such a thing as flu virus in the sense of a monolithic disease vector existing outside of us, conceived as it is as the relationships of predator to prey? First, consider that the highly authoritative Cochrane collaboration acknowledges that there are many different flu viruses that are not in fact influenza A, against which flu vaccines are targeted, but which nonetheless can contribute to symptoms identical to those attributed to influenza A. Over 200 viruses cause influenza and influenza-like illnesses, which produce the same symptoms: fever, headache, aches and pains, coughs and runny noses. Without laboratory tests, doctors cannot tell the two illnesses apart. Both last for days and rarely lead to death or serious illness. At best, vaccines might be effective against only influenza A and B, which represent about 10% of all circulating viruses. Source: Cochrane Summaries. Emphasis added. This makes for a picture of complexity that powerfully undermines health policies that presuppose vaccination equates to a bona fide immunity, and by implication necessitates the herd collectively participate in the ritual of mass vaccination campaigns as a matter of social responsibility and necessity. Even the use of the word immunization to describe vaccination is highly misleading. The moment the word is used, it already proposes efficiency, and it makes it appear as if non-vaccinators are anti-immunity. Instead, what they actually are pro-immunity via clean air, food, water, and sunlight, but unwilling to subject themselves or their healthy children to unavoidably unsafe medical procedures with only theoretical benefits. Why flu viruses doesn't exist the way we were told. But the topic gets more interesting when we consider the findings of a 2015 study entitled Conserved and Host-Specific Features of Influenza Virion and Architecture. This was the first study ever to plumb the molecular depths of what influenza virus is actually composed of. Amazingly, given the long history of vaccine use and promotion, the full characterization of what proteins it contains and what they are derived from was never previously performed. How we invest billions of dollars annually into flu vaccines and have created a global campaign to countermand viral enemy whose basic building blocks we don't even know. It's hard to understand, but it is true nonetheless. The study abstract opens with this highly proactive line. Viruses use virons to spread between hosts, and viron composition is therefore the primary determinant of viral transmissibility and immunogenicity, emphasis added. Virons are also known as viral particles, and they are means by which viral nucleic acids are able to move and infect living organisms without the viral particle taxi to carry around the virus DNA passenger. It will be harmless. In fact, viruses are often described as existing somewhere between living and inanimate objects. For this reason, they do not produce their own energy, nor are transmissible without a living host. And so in this first line, the authors are making it clear that viron composition is also the primary determinant in how or whether a virus is infectious, transmits, and what effects it will have in the immune system of the infected host. Influenza viral particles. This distinction is important because we often think of viruses as simply pathogenic strings of DNA or RNA. The irony, of course, is that the very things we attribute to so much latency to viral nucleic acids are not even alive and cannot infect an organism without all the other components, proteins, lipids, extraviral nucleic acids, which are technically not viral in origin, participating in the process. And so if the components that are non-viral are essential for the virus to cause harm, how can we continue to maintain that we are up against a monolithic disease entity out there who infects us, a passive victim? It's fundamentally nonsensical given these findings. It also clearly undermines the incessant fear-based rhetoric those beholden to the pro-vaccine stance to cohere the masses into undergoing the largely faith-based rite of vaccination. Let's dive deeper into the study's finding. The next line of abstract addresses the fact we opened this article with, namely that there is a great complexity involved at the level of the profound variability in viron composition. However, the virons of many viruses are complex and phylomorphic, making them difficult to analyze in detail. But this problem of the great variability in the viron composition of influenza is exactly why the study was conducted. They explain here we address this by identifying and quantifying viral proteins with mass spectrometry, producing a complete and quantifiable model of the hundreds of viral and host-encoded proteins that make up the phylomorphic virons of influenza virus. We show that a conserved influenza viron architecture, which includes substantial quantities of host proteins as well as the viral protein NSI, is elaborated with abundant host-dependent features. As a result, influenza virons produced by mammalian and avian hosts have distinct protein compositions. In other words, they found that flu viruses is as much comprised of biological material from the host the virus infects as the viral genetic material of the virus per se. How, then, do we differentiate influenza virus as fully other, given that it would not exist without self-proteins, or those of other host animals like birds, avian, or insects, that would be impossible to do with any intellectual honesty intact? There's also the significant problem presented by flu vaccine production. Presently, human flu vaccine antigen is produced via insects and chicken eggs. This means that the virus particles extracted from those hosts would contain foreign proteins, and would therefore produce different and/or unpredictable immunological responses in humans that would be expected from human influenza viral particles. One possibility is that the dozens of foreign proteins found within avian influenza could theoretically produce antigens in humans that cross-react with self-structures resulting in autoimmunity. Safety testing presently does not test for these cross-reactions. Clearly, this discovery opens up a Pandora's box of potential problems that have never been significantly analyzed. Since it was never understood until now that influenza was so thoroughly dependent upon a host for its transmissibility and immunogenicity. Are flu viruses really hijacked exosomes? Lastly, the study identified something even more amazing. Finally, we note that influenza virions share an underlying protein composition with exosomes, suggesting that influenza virions form by subverting microvesicle production. What these researchers are talking about is a discovery that virion particles share stunning similarities to naturally occurring virus-like particles produced by all living cells called exosomes. Exosomes, like many viruses, i.e. enveloped viruses, are enclosed in a membrane and are within the 50 to 100 nanometer size range that viruses are, to 20 to 400 nanometers. They also contain biologically active molecules such as proteins and lipids, as well as information containing ones like RNAs, exactly or very similar to the types of contents you find in viral particles. When we start to look at viruses through the lens of their overlap with exosomes, which are carriers of RNAs, are essential for regulating the expression of the vast majority of the human genome, we start to understand how their function could be considered neutral as information carriers, if not beneficial. Both exosomes and viruses may actually be responsible for interspecies or cross-kingdom communication and regulation within the biosphere, given the way they are able to facilitate and mediate horizontal information transfer between organisms. Even eating a piece of fruit containing these exosomes can alter the expression of vitally important genes within our body. Exosomes. In light of this post-germ theory perspective, viruses could be described as pieces of information inserts of chromosomes, not inherently bad, but in fact essential for mitigating the genotype/phenotype relationship with organisms, who must adapt to ever-shifting environmental conditions in real time in order to survive, something the glacial pace of genetic changes within the primary nucleotide sequences of our DNA cannot do. For instance, it may take 100,000 years for a protein-coding gene sequence to change versus seconds for a protein-coding gene expression to be altered via modulation via viral or exosomal I guess they're pronounced it RNAs. This does not mean they're all good. Either sometimes, given many conditions outside their control, their messages could present challenges or misinformation to the cells to which they are exposed, which could result in a disease symptom. These disease symptoms are often, if not invariably, attempts by the body to self-regulate and ultimately improve and heal itself. In other words, the virion composition of viruses appear to be the byproduct of the cell's normal exosome, also known as microvesicle, production, machinery, and trafficking, albeit being influenced by influenza DNA. And like exosomes, viruses may be a means of extracellular communication between cells instead of simply a pathological disease entity. This could explain why an accumulating body of research on the role of the virome in human health indicates that so-called infectious agents, including viruses like measles, confer significant health benefits. See the health benefits of measles and the healing power of germs. Other researchers have come to similar discoveries about the relationship between exosomes and viruses, sometimes describing viral hijacking of exosomes pathways as a Trojan horse hypothesis. HIV may provide such an example. Concluding remarks. The remarkably recent discovery of the host-dependent nature of influenza viruses, viral composition, is really just a tip of an intellectual iceberg that has yet to fully emerge into the light of day, but is already sinking ships, paradigm shifts, if you will. One such paradigm is that germs are enemy combatants and that viruses serve no fundamental role in our health and should be eradicated from the earth with drugs and vaccines if possible. This belief, however, is untangible. With the discovery of the indispensable role of the microbiome and the subpopulation of viruses within it, the virome, we have entered into an entirely new, ecologically based view of the body and its environments that are fundamentally inseparable. Environs that are fundamentally inseparable. Iconically, the only thing that influenza may be capable of killing is germ theory itself. Hope you enjoyed the write-up, everybody. It'd be great if you subscribed. Awesome if you leave me with some comments. Thanks a lot, everybody. Bye.